For the first time, the genome of the cultivated tomato, Solanum lycopersicum, has been decoded. This is the culmination of nearly 10 years of work by the Tomato Genome Consortium, an international collaboration of over 300 scientists. This work is an important step toward improving yield, nutrition, disease resistance, taste, and color of the tomato and other crops.
I'm tremendously proud of the contributions that Boyce Thompson Institute scientists have made to this very large project. In fact, scientific projects these days get larger and larger and require more and more different kinds of expertise to come to fruition. Sequencing the tomato genome involved more than just taking DNA from a tomato fruit and determining its composition. In this team, leadership was particularly important because the collaboration involved scientists from around the world. Joyce Van Eck and Jim Giovannoni both had key roles in moving the collaboration forward, keeping it together over the past 10 years or so, and bringing it to a successful conclusion. In addition, understanding and the meaning of the DNA once it's decoded is a very complex computational problem. After all, tomatoes have over 35,000 different genes, more than people. Zhangjun and Lucas Mueller both played key roles in making this information understandable and accessible to scientists around the world. The real value will come in the next years as that information is accessed and put to use.
Join us as we talk to a few of the consortium scientists from the Boyce Thompson Institute for Plant Research at Cornell University to discuss their involvement on the project and what it means for tomato shoppers everywhere.
Everybody's familiar with tomatoes. Everyone who's grown them in their garden knows what a good tomato tastes like. And everyone who's bought one at the supermarket knows the potential that's there for good flavor that's often missing. From the perspective of improving tomato, it's an incredible resource. We now essentially have all the genes captured. We know their sequences, and now the fun part of understanding what they all do begins. But even without knowing exactly what they do, breeders and geneticists will be able to use that information to improve the quality and production characteristics of tomatoes. Tomato is related to a lot of other important vegetable crops, things like pepper, eggplant, potato. These are all members of a family called the Solanaceae. And with the tomato genome sequence and information from some of these other genomes, we can now see exactly how closely related those different genomes are. And we can actually use information from tomato, for example, to assist breeding of other crops from this related family, the Solanaceae. One of the interesting features observed by comparing the tomato genome sequence to other previously sequenced plant genomes is that there's evidence for what's called a triplication event in one of the ancestors of the current tomato. And that triplication means that the genome of that ancestor was multiplied 3 times. Now, why would a triplication occur? Well, it turns out that that event correlates very well with the timing of the last mass extinction on Earth. And normally an event like a triplication of the genome or a large rearrangement or change in the genome wouldn't be tolerated very well. But in that kind of extreme environment, perhaps it was, and it was selected for. Now, since that event, many of those genes have been lost. But one of the interesting things about the tomato genome sequence is that some of the genes that remained are genes that are very important for how the fruit matures and develops and looks today. So in 2003, a group of tomato researchers got together with the goal of developing tools that would assist in understanding tomato biology and improving the tomato crop. And in 2009, the first draft genome sequence was released, and that was released through the Sol Genomics website.
The SGN database is a large database for tomato and Solanaceae genomics, and is the central point where all the information from the project flow together and then is accessed by researchers around the world to get to the tomato genome information.
Not only did we have funding to sequence the genome, but we also had funding to do educational outreach. I always believe in starting out, getting them young, and having activities for groups in kindergarten through 5th grade, and middle school, and also in high school. I put together a newsletter. So the purpose of the newsletter was to be able to keep everyone connected within the project. And that grew from just talking about the sequencing updates to then having a whole community resource where we talked about other genome updates. We even had Solanaceae recipes in there. Currently we have around 800 people worldwide on this newsletter list.
So genome sequences are never really finished, but at some point you have to publish it and release all of the information. And so once the genome reached what we believed, the consortium believed, was a very high quality, where most, if not virtually all of the genes were captured, most were assigned a position on chromosomes, and much of the non-genic DNA was also defined, we chose to publish at that time.
So my group mainly using all the existing tomato resource and try to evaluate the quality of the tomato genome. The quality is very high.
Recently, there was a breeders' roundtable meeting in Ithaca for tomato breeders, and the interest of the breeders in the tomato genome were enormous.
And one of the real important features of the genome sequence is that it's fully publicly available. So the groups that developed the sequence were doing so with funds from their respective governments, with the intention of making this information available to every scientist, every seed company, everyone who would be interested in using that genome sequence for the betterment of tomato products and ultimately human health and nutrition.
The sequence is made available without any strings attached, completely free to everyone who wants to use it.
We had researchers from all over the world working towards a common goal of generating a high-quality tomato genome sequence, and that we worked very well together.
I think we needed to generate a very high-quality genome sequence. This is very helpful for downstream analysis, also for breeders, for researchers.
I think for the consumer, the main value out of this project is that it's going to lead to better fruit and vegetable products.
Tomato genome and sequencing research in the United States was supported by the National Science Foundation and the U.S. Department of Agriculture. The Boyce Thompson Institute is a private, nonprofit research institute situated on the Cornell University campus that explores fundamental aspects of plant biology to positively impact society.